Our presentation tonight is a review of 3 species level taxa of the Anthocharis Sara complex, the Sara orange tip, by Todd Stout. Todd has over 35 years experience studying and raising butterflies in many portions of the US, and he has provided school and public presentations and displayed exhibits on butterflies and their life cycles. Along with several other lepidopterists, Todd is currently working on a book on the butterflies of Utah. He has served as past president and secretary of the Utah Lepidopterist Society and has served on the executive council of the Lepidopterist Society. And he was the MC for the Lepidopterist Society 2014 meeting held in Park City, Utah. He is currently a University of Florida research associate, and he described a new subspecies of the Julia orange tip in western Colorado in 2012. In 2018, he published a taxonomic revision of the Sara orange tip, consisting of 3 species in western North America based upon 5 character sets. And those are adult phenotypes, larval coloration, pupal cone angle, overwintering cycles, and species level contact zone behavior. He's also co-authored a paper, DNA barcodes reveal different speciation scenarios in the 4 North American Anthocharis. So Todd has wanted to give back to the next generation of lepidopterists, and he created the Utah Bug Club program in 2004 and worked with Bob Pyle and several other organizations teaching children and families about butterflies and getting nets into the hands of kids. So that is fabulous for our future. He continues to provide butterfly education by founding Utah Butterfly Field Trips on Facebook, which has been active since 2008. He created the Raising Butterflies DIY website in 2010, which teaches how to locate and raise many North American butterflies. He also owns a business providing butterfly breeding equipment under the same brand. Thank you so much for being with us tonight, Todd. So I see you shared your screen, so you can go ahead and get started.
Sounds great. Hey, thanks for inviting me here. It's a great opportunity. I probably didn't need to include in my bio about the paper because that's what I'm going to be reviewing tonight. So I'm glad to be here. I wish I were with you in person because I like giving PowerPoint presentations in person where I can interact with the audience. They can ask questions at will. But I have done some PowerPoint presentations through Zoom, so here we go. I started in the late 1990s studying orange tips when I noticed consistent larval differences between 2 varieties in Utah, which got me going. I need to put my cursor on this to advance the slide. So let me see, let me just got it here, just a sec. Okay, so this paper can be downloaded on the bit.ly link that you see on the right of the screen, and it can also be downloaded from wildbutterflyassoc.org. My paper was written and published on March 30th, 2018 in Insecta Mundi and is available online, as I said before. Okay, this presentation explores the arrangement of 3 species, Sara, Julia, and southwestern orange tips, based upon a combination of adult wing patterns, 5th instar larval, 5th instar caterpillar coloration, shape of the cone of their pupae, number of overwintering cycles of their pupae, and lastly, and the most granular we'll get into is contact zone behavior. How do they behave when they fly in sympatry? And sympatry means simply when they fly together. Before diving into the particulars of these 5 character sets shown in the previous slide, I'm going to spend a few minutes sharing several slides to establish a baseline of the basics of this presentation, starting with some terms. The Anthocharis Sara complex, or A-Sara complex, is simply a blanket term referring to all 3 species in the group that I'll be talking about. I may make the mistake of saying Sara orange tips when I mean the group, not just necessarily Anthocharis Sara, but I'll try to keep it to Anthocharis Sara complex or A-Sara complex when I'm just talking about the group of 3 species. And number 2, larvae feed on mustards of the family Brassicaceae, including many rock cresses. Sara orange tips, speaking of the group, with few exceptions, basically have one spring flight per year, where eggs hatch in about 5 days and larvae complete their development in less than a month. We'll be talking about the wing differences between different varieties of orange tips, but we'll also be talking about sex differences between males and females, both in behavior and in wing patterns. During this presentation, I may mention the term type locality or topotypes. Type locality simply means a location where a butterfly was originally described, and topotypes simply are specimens studied from the type locality. When two different species of orange tips fly together, I might use the term sympatric or synchronic, which simply means that they're just flying together at the same time and at the same place. In order to compare apples to apples for this presentation, the lion's share of adult photographs, unlike the photo above, will show specimen photos of pinned adults, the majority of which were raised from caterpillars for this research. I also provided extensive immature butterfly photography of caterpillars and pupae, which are central to the data provided for this research. A central theme to this presentation, I can't overemphasize this enough, simply is using wing markings by themselves to differentiate these three species from each other is problematic, as there is overlap in wing markings amongst all three spanning the entire complex. I do not mean to discourage the use of field guides for Sara orange tips, speaking of the complex, but knowing your own location when you see an orange tip in the field is far more helpful than looking at a field guide with diagrams pointing to the wings. And all I mean by that is these diagrams tend to be overgeneralized. Throughout my presentation, I'll be sharing this hand-drawn distribution map of the subspecies of these three species. I realize that these maps aren't as nice as Caitlin LeBar's maps that are generated by ArcGIS. Someday I hope to be able to learn that tool. Also, I realized that this map is kind of busy, so I wanted to break it down into the three separate maps reflecting the subspecies of the three species. Because if you look at this map, I know it's really busy, showing all the subspecies. So here are three separate distribution maps of the Julia orange tip on the left. In the middle are Sara orange tips and southwestern orange tips. During my presentation, you may hear me say thoosa, southwestern. Julia is pretty specific. So if I say Julia, I mean Julia orange tip or Anthocharis Julia, Sara orange tip, Anthocharis Sara. Sometimes I go back and forth between scientific names and common names, but southwestern may be a little confusing if I keep saying thoosa. Thoosa equals southwestern orange tip. So if you look at their basic distribution, you can see that the most expansive is the Julia orange tip. Sara orange tip is basically a California endemic, and southwestern is basically an orange tip from the desert Southwest, Great Basin, and Colorado Plateau regions of Western North America. So in terms of distribution, if we break down the distribution of these three species, think of orange tips as a boreal forested butterfly that flies in zone 3 of this map, in the northern portion of Western North America, from the panhandle of Alaska south through Western Canada and through the Pacific Northwest. It then continues south to zone 2 into the northern half of California and the Western US. Conversely, and contrastedly, Sara and southwestern orange tips are just the opposite and are exclusive to zone 1, meaning you won't find Julia in zone 1, and fly in the southern portion of Western North America from Mexico into Southern California and the Southwest, continuing into zone 2, approaching and just barely extending past Oregon's and Idaho's southern border. So if it gets a little confusing, just think of Julia as the northern bug, Sara and thoosa are the southern species, and zone 2, right there in the middle, that's where things get interesting, where they can overlap and where they can meet. What this means for the evergreen state is that only Julia orange tips fly in Washington. Some of these maps will be confusing, but if you are in the evergreen state and you see a Sara complex orange tip, it's Julia. The yellow-orange region in this map is a broad generalization of the Cascades where the two subspecies meet. Anthocharis Julia sulphuris, that's the name of the subspecies, east of the Cascades, and west of the Cascades is Flora. Now, this map is somewhat flawed in that yellow-orange that you see in this map is a generalization of the Cascades where they meet, and John has told me, you guys know more than I do as to where you can find intergrades between these two subspecies. This is just a broad generalization for the state of Washington. A previous taxonomic arrangement of the Sara complex in Washington had Sara orange tips themselves flying in Washington. This is John Hinchliff's 1996 map found in an atlas of Washington butterflies showing subspecies flora west of the Cascades. and what was called A. serrastella, Anthocharis serrastella, east of the Cascades. However, based upon my research, looking beyond adult wing characters exclusively, Sara orange tips, or Anthocharis sara, are basically a California endemic comprising of the 4 subspecies you see in the map, where the nominate subspecies extends into southwest Oregon from Baja California. By the way, when I say nominate or nominotypical, I mean whenever a butterfly species is described, where maybe a new subspecies was later described, the first subspecies carries the name of the species. In other words, nominate Anthocharis sara is Anthocharis sara. The first subspecies of the monarch is Danaus plexippus plexippus. It's when the species and the subspecies are the same. That's what nominate or nominotypical means. Also, Stella is now considered a subspecies of the Julia orange tip and is restricted to these central California counties surrounding Lake Tahoe and barely extending into western Nevada. The differences between, and here's just a photo showing some specimens of Anthocharis juliiflora west of the Cascades, contrasted to Anthocharis juliisulphuris east of the Cascades. Also, when you look at Sara orange tips, you can see that these are quite different. You have male on the left and female on the right, where females of some populations are yellow, whereas others are not. And most of the females for Washington Julia are yellow. But you can see the dark markings are more significant west of the Cascades with flora than they are east of the Cascades with sulphuris. This is my attempt to be funny. So you guys can blame me and John Pelham, who named sulphuris, for the shipping of Sara and Stella orange tips back to California, where they belong. Anyway, one of the wing characters I'll be referencing today as we talk about the differences in orange tips is illustrated by the black forewing apical border. This border mostly surrounds the orange tip, and on the males tends to be solid black, whereas on the females, the dark markings, which is called the forewing black apical border or black forewing apical border, is checkered. So if you just look at the male and the female, you can see the basic sex differences, and then the black apical borders also are different males from females. Also, another feature that I'll be referenced are the forewing discal cell bars, as shown in yellow. In some adults of Sara orange tips, the forewing black apical border connects with the discal cell bar, as applicable to the male on the photo on the left. With others, it doesn't connect, as is the case with the female on the right. And so we're going to be talking, as we get into the contact zones on the wing features, the connecting or disconnecting between the forewing black apical border and the discal cell bar. Also, with some Sara orange tips, and I'm going back to the complex, you'll get these dorsal hindwing black marginal spots that are very strong in some specimens and weak in others. And then on the underside of the butterfly, we'll be talking about the ventral hindwing modeling. Is it strong? Is it weak? Is it green? Is it gray? And so on and so forth. So for my research, I studied over 90 populations spanning all 50 named subspecies. Personally, I traveled to 10 Western US states and described new subspecies of A. julia, as we said before. And I visited 12 of the 15 type localities in the Western US. Here are some photos of the different mustards and rock cresses that Sara orange tip complex caterpillars use in nature. In Utah, if you look at the bottom mustards there, you'll see dyer's wood, which is really helpful. Dyer's wood is really frowned upon by naturalists in our state because of how invasive, it's non-native and invasive, but what was nice for the research is whenever I found caterpillars, I didn't always have to go into the mountains to take cuttings of rock cresses. I would use dyer's wood, and all 3 species would do just fine on it. So that's kind of one of the ironies. I'm not going to go into a lot of detail about rearing members of Sara orange tips, but in order to obtain early stages, I basically had 2 options because I did a lot of rearing studies, obviously, if I didn't make that clear earlier, I did a ton of rearing studies with this group. And the 2 options of studying the early stages are either finding eggs and caterpillars in the field or collecting females and setting them up to lay eggs, and then separating out those eggs, as they can be, little first instars can be cannibalistic, and then raising them that way. So this photo you see here is a rock cress in bloom, and this is ideal when you're looking for eggs or for caterpillars. And if you look on the left of this diagram, you see a blown up image of an egg. When you find these orange eggs on mustards, the different types of whites, marbles, have preferences of where they like to lay their eggs. For example, the marbles, as you can see circled on top, lay their eggs on the buds of these rock cresses, whereas you see the Anthocharis complex is circled. That's a pedicel part of the plant. And then down below, you'll see possibly multiple eggs of spring whites. So location means something if you want to be able to separate marbles, orange tips from spring whites. Here's a beautiful photo of a female Sara orange tip laying an egg on a pedestal of a rock crest. This picture was taken by Dennis Walker and used with permission. What's interesting is the exception to the rule. When I say there's preferences of where eggs are like to be laid, that's not always the case. You see an egg here on the stem, but it's off-white, suggesting that also is a Sara complex. And sometimes they will break their own rules. I saw that when I did studies of Anthocharis Julia Stella around Lake Tahoe. Some of the mustards had their heads chewed off. And so the females adapted by laying eggs on the stems. And so in this picture, you see the egg laid there. I don't want to spend too much time. This graphic is a little bit busy. But when we talk about orange eggs, those are the fertile eggs. If you happen to find an egg that's not orange, that's cone shape, you can also separate it by species or groups by the color. Sara orange tip eggs, the complex, tend to be off-white. The large marbles tend to be ivory white. It's not really shown well in this graphic, but in the field, it'd be on a bud too. So I could tell the difference between a large marble egg and a Sara orange tip complex egg. The desert marble eggs are light blue, also on the bud. And of course, the spring whites are a yellowish green. If you're looking for caterpillars, there's also the difference of caterpillars of these 4 pyres as shown in this slide. To raise them, I would simply put cuttings in a water bottle and put that in a bucket or a pop-up cage and replace out the cuttings about every week or so. And they would go from egg to pupa in about 17 days. Once they form a chrysalis, I place them in a tub in a dark closet during the summer months where they are overwintered either outside a protective enclosure or in the refrigerator with humidity from November until late winter. So this is how they would be overwintered. And one thing I'll be talking about later is they don't always emerge after one overwintering cycle, especially if they fly in a xeric area. They can extend hibernation for 2 or 3 years. That probably does not happen in Washington because of all the rain you get. I realize Western Washington gets a lot more rain than Eastern Washington, but I did notice that in the more xeric areas of the Great Basin, especially in our deserts, the pupae would extend hibernation for multiple years. Okay, and for my research, I did keep records on spreadsheets. This is just a screenshot of one of those spreadsheets with all of the particulars. Let's go back to talking about male versus females, wing patterns. I'm sure many of you have seen males patrolling mountain canyons, ravines, or ditches on adjacent roads or trails in search of females. This photo was taken in Thoosa Canyon, California, where male Sara orange tips fly. This is another picture taken in Utah, in Juab County, where both southwestern and Julie orange tips patrol together. This is one of the contact zones between 2 species I'll be talking about here in a little bit. And I talked about this before, but obviously males and females are different. I just want to do a quick, this is Nevada, subspecies pseudothusa of Sara. Here's the ventral of that butterfly. Here's Anthocharis Julia alaskensis. Even though that looks differently from Sara pseudothusa, you can still see the obvious differences between males and females. Here's Northern California Anthocharis Sara sempervirens, both yellow, but still these males versus female wing pattern difference seem relatively consistent, even though they change from variety to variety. Here's Washington Julia flora and Julia sulphuris. So before we talk about the adult characters, I'm still going to be talking about the 5 character sets. I want to do a quick run around for the 15 subspecies. I can't give the full variation, but I just want to do a quick overview of all the different subspecies of the 3 species. So let's start with Sara orange tips. A lot of the variation that you're seeing here is on a population level, and these are topotypes. So the type locality of Sara is in Queen Lily Campground in Plumas County, California. And if you look on the females on the right, this is the amount of variation you're just getting just from the type locality. They have a white female, a yellow female with less black markings. The last yellowish female oddly looks, now I'm jumping to Anthocharis Sara sempervirens. Like I said, I'm going to go through this kind of rapidly because we're short on time. Kind of looks like the Sara sempervirens that I raised from Northern California in Siskiyou County. So here's some more variation. If you looked on the males, the second one is a yellowish male. And if you look at the females, on the bottom, you got a whitish female, but the rest are female. So when we talk about variation of orange tips, there's three tiers basically, upon a population level, which can be significant, a subspecific level, which is an aggregation of all the populations. And then we take a look at all the subspecies within a species. I mean, there's just a lot of room for variation just for a single species. And that's why I said earlier that drawing conclusions based upon wing characters alone can be problematic because all three species can vary so much. Here's another subspecies of Sara called pseudothusa that flies around Lake Tahoe, where the females are mostly white, except for the last one on the right, you see that she's yellow. Males are pretty consistent. There's another subspecies of Sara in Catalina Island off of the coast of Southern California. And Santa Cruz Island, it's called Anthocharis saragunderi. The black markings tend to be a lot more extensive. If you look at the third male on the left side, that almost looks like a southwestern orange tip. But if you look at the underside marbling or modeling, it's green. And so that's what Sara can do sometimes in the first generation. So that's a fast overview of some of the topotypes of the four subspecies of Sara. Let's jump into Julia. In northern Utah, we have one of the most washed out subspecies called Anthocharis Julia browningi. And your subspecies in eastern Washington is so expansive that it extends from eastern Washington to Idaho and dips into Utah. So this is Anthocharis Julia sulphuris. This is where Utah and Washington kind of combine. We both have this subspecies. The previous subspecies flies in the Wasatch, whereas Julia sulphuris, did you have a question? I'm sorry. Julia sulphuris flies in the high Uintas and on the eastern portion outside of our Wasatch Range. And those are the two subspecies shown side by side. Where browning eyes just tends to be a lot more washed out than sulphuris, and sulphuris is more washed out than flora in the state of Washington. John Pelham named Anthocharis Julia sulphuris in 2008 from Shoshone County, Idaho, near Kellogg. And so I drove up there. This is in the panhandle of Idaho, about 80 miles east of the eastern border of Washington. And so I wanted to be able to study the topotypes of Julia sulphuris where it was described, where John described it. So there's some of the variation from that type locality. And more pictures of western Washington flora. This picture shows a topotype from Tenino, Thurston County, Washington, of flora. But this photo was taken at the McGuire Center, and my camera settings were somewhat flawed. I took all these pictures when I went to the McGuire. I'll talk about that here in a little bit. But you can see the hue of yellow is not realistic. And so, but this was taken, this photo was taken, like I said, from the McGuire Center. We talked about Anthocharis Julia. We're talking about Anthocharis. Somebody needs to mute.
That's out all the
Anthocharis Julia Stella.
Let me stop for just a moment, Todd. If you're not muted, would you please do so? There's a little bit of background noise, so thank you. Go ahead, Todd.
Keep talking, Paulette. I'm just going to take a quick drink of water.
Okay.
I'm talking so much, my mouth is dry. I wish I were listening than talking. Anyway, the subspecies Stella flies around Lake Tahoe into eastern Nevada. And what's interesting is you see on the left, you have yellowish males. And this is Anthocharis Julia alaskensis. And I want to give a quick plug to the late Canem Philip because I was able to study the immature stages of this through a great sacrifice by Ken Phillip. I'll talk about that towards the end of my presentation. But he was a rock star in getting me females so that I could get eggs, so that I could take a look at the early stages of Anthocharis Julia alaskensis. And this is a photo that he took and sent me. He actually sent me this female. Jumping over Utah into Colorado, this is nominotypical. Again, the species and the subspecies is the same, Anthocharis julia of the Julia orange tip from the Front Range. sometimes with this population in the Front Range, you'll get white females of Julia, but normally they're yellow, as you can see in the upper right-hand corner. For all these pictures, I got males on the left, females on the right, dorsal above, ventral below. I try to stay consistent. And I want to talk, Bob just talked about the Pacific Northwest, sorry, getting tongue twisted, I'm talking so fast, trying to get through all these slides. We talked about the Pacific Northwest Lepidoptera workshop of 2005, which I got to attend, and I did another presentation on Sara orange tips, much more limited. See a lot of people that we know. I see Bob, Floyd, and June Preston. I see Andy and John in the back, my friend Abe Holman, and several other people. But I want to talk about Floyd and June Preston. I became reacquainted with them during this meeting. They're wonderful people, salt of the earth. I'm so glad that I got to meet them on a couple occasions. But they came to visit me on the 31st of May, 2007. And they had just been collecting orange tips in Colorado. And Floyd came up to me, and he didn't show me these actual specimens, but he collected and pinned some orange tips of the Sara complex in Grizzly Creek near Glenwood Springs. He says, "Todd, are these thoosa or Julia?" And I looked at him, I said, "Oh, those are thoosa, those are Julia. What the heck are those?" I mean, I was blown away. This is a population at that time in 2007. I had no idea that these phenotypes that seemed to look like Julia and thoosa were coming up. My background was to compare them to my Julia is browning eye. Do they look like browning eye? No. The dark markings are more extensive. And then I've got thoosa, which have heavier dark markings than this population. So the next day I drove out there in hopes of finding males and females. And I think it was bad weather, but I went there after other years to collect females, to find eggs and larvae, to rear them out, to try to assign them to thoosa or Julia, because looking at the adult characters, I was blown away. And so I ended up, when I studied the caterpillars and the pupae, they were clearly Julia and not thoosa. And since previous publications had these assigned to as southwestern orange tips or thoosa, I named a new subspecies, because clearly these are not Julia, and they're not species thoosa. So I started studying these populations in the counties west of the Continental Divide and named it Julia prestanorum after Floyd and June. And I named it in the Butterflies of Colorado, Part 5, Papyridae and Papilionidae by Michael S. Fisher. And this was in 2012. And it was just, these two people, they've since passed away, but they made a t-shirt in honor of the name. I know Bob named a butterfly after June, and when we go and share, Bob, if you could tell us what you named after June, because it's escaping my memory right now. But that was a wonderful opportunity to name a butterfly after them. Let's talk briefly about southwestern orange tips. Like Sara, there's 4 subspecies. In Utah, we have thoosa. Type locality is just south of Utah border in extreme northern Arizona, and the dark markings are significant. And as Mike Fisher named Anthocharis thoosa Colorado along with Jim Scott, and this is thoosa. Thoosa Colorado, the discal cell bars are much thinner, both on the males and the females. I keep going back and forth between thoosa, and thoosa Colorado. And on my thoosa female, on the upper right-hand side, you can see the discal cell bar is connected to the black apical border, whereas this female on Colorado is not. So I think thoosa Colorado is a good subspecies that Mike Fisher and Jim Scott named. If you go south of Utah, down towards Tucson and Phoenix, Arizona, another subspecies of the southwestern is called Anthocharis thoosa ingami. And here's just a comparison of thoosa from Utah with thoosa ingami from southern Arizona and central Arizona. And another subspecies similar to Colorado, virtually indistinguishable, is Coriandii, also named by Jim Scott. Okay, so let's start talking about the 5 character sets that I introduced. So basically, like I say, the 5 character sets are adult phenotypes, 5th instar level coloration. We've talked a lot about the adult phenotypes. of these 5 character sets, no singular character is airtight. For example, part of the problem with previous taxonomic reviews of this butterfly is using adult phenotypes by itself has led to incorrect conclusion. Fifth instar larval coloration is more of a solid way to separate the species, but even that has an exception in Northern California and in Southwest Oregon, where Sara orange tips aren't as green as the rest of all the subspecies. They tend to get lighter like thoosa. The third one, pupal cone curvature and color, is great in separating southwestern orange tips from Julia orange tips, but is not consistent for Sara. Number of overwintering cycles and interspecific contact zone behavior, we'll talk about that when we reach them. So when I met with Andy Warren and was doing this, Andy basically told me 2 things. He says, for taxonomic work, use as many character sets as you can to document evidence, because there for a while, all I was looking at was larval characters. And he also told me when analyzing the attributes of any subspecies, start with topotypes. You know, if you're going to study Anthocharis juliasulphuris, go where John named it in northern Idaho, in Kellogg in Shoshone County. Okay, I hit the wrong button. I'm back to John. So this picture is pixelated, but that was the advice that Andy gave me. So these represent the type localities of all the subspecies of all the species. And out of 15, I personally visited 12. Anthocharis juliiflora, I didn't in your state, but I do have photos that I analyzed from Tenino, Washington. Julia alaskensis, I didn't go to Alaska. Ken Phillips saved my hind there. And Anthocharis sempervirens, I got help from Ken Hansen. Getting Ken Phillips and Ken Hansen. Ken Hansen got larvae for me and eggs outside of Redwood National Park, where I was able to study those. So as I said before, drawing conclusions using, well, I'll go ahead and read this. This is actually right out of my paper. It says, as mentioned, the historical treatment of the Sara complex relied heavily on adult morphological characters to infer species level distinctions. This is problematic because overlap in adult morphological characters of all 3 species make it challenging to identify wing traits exclusive to any single species. And then I go into specifics. And so in that which is highlighted in yellow, I'm just going to bypass that because I actually showed examples of this. I came up with a generalization of character, dorsal white males and white females, and showed examples of all 3 species that show that trait. Same thing with dorsal white males and white yellow females. I showed examples of all 3 species and so on and so forth going down in this slide. The next slide shows males. And here's a character set up on top, upper left. It says male dorsal discal cell bar is offset and disconnected with the forewing black apical border. And I showed example of all 3 species where that character trait applies, and so on and so forth. You can read these. This is right out of the paper. And so I'm just trying to, solidify my point. If you're only using adult characters, it's going to be difficult. And one thing Jim Scott said in one of his papers, and I agree, is if you're going to talk about the differences between species, do it on a local level. And we'll be talking about that in the contact zone. So in order to further my education on the adult characters, I was really grateful for Andy Warren, who flew me out to the McGuire Center for Lepidoptera and Biodiversity in Florida, where I spent 3 days staring at tens of thousands of Sara complex orange tips. And Andy told me, if you're going to be at the McGuire Center, you've got to stand by the eastern black swallowtail that's nectaring on your knee. So I took that picture. So anyway, here's just a picture of me being blown away by all the curating that Andy did at the McGuire Center. I ended up taking 862 photos in 3 days of all the subspecies, tray after tray. I'm not going to drill into the details of all these. Most of these are Sara, most of these are Stella. But I just stared at them, took down notes, took down photographs, put them in a spreadsheet to try to solidify, what I was saying with my previous research. So let's jump into the second parameter, fifth instar larval coloration. There are basically 3 basic color patterns amongst fifth instar larvae. In general, these are dark green for Sara, medium green for thoosa, and light green for Julia. Now that's a little overgeneralized, but I'll show specific examples in the larvae here coming up. So how did all this get started? what got me started noticing fifth instar larval differences happened pretty much in my backyard in northern Utah. I spent my college years in Provo, BYU, and later moved an hour north to Salt Lake City. Both Utah and Salt Lake valleys literally are a buffer between the western limit of the Rocky Mountains and the East Wasatch Mountains, and the eastern limit of the Great Basin or Basin Range province to the west, where both flora and fauna tend to be different. So I kept noticing how different our mountains were. The Ochre Mountains, the Stansbury Mountains, were part of the Basin Range province, and to the east, the Wasatch Mountains are part of the Rocky Mountains. I know Utah is an interesting state, and this is the political boundaries, but from a geological standpoint, I almost feel that Nevada is 10 miles west of me and Colorado is 5 miles east of me, because these two habitats are completely different. And so I was really interested in studying these two orange tips because they're very diverse in how they look. And as I raised them, I noticed that the Julia orange tips, subspecies browning eye, were a lighter green than the southwestern orange tips, Anthocharis thoosa. And then when I expanded my research and went to southern Arizona to raise what we call Anthocharis thoosa ingami, those larvae look like thoosa from Utah. But when I went to Boise, Idaho and raised sulphuris, those larvae look like Julia browning eye. So Julia and thoosa began to have consistently different caterpillars. And what I'm basically saying is, here's a picture of a Julia caterpillar. Basically, this photo is taken 48 hours after it molted to fifth instar. And if you look closely, the white lateral stripe, I'll be mentioning that quite a bit, is contrasted to the green color of the rest of the caterpillar. It's a very subtle transition. And this is true with all the subspecies of Julia. Whereas with thoosa, if you look closely at the white lateral stripe, it's not quite as stark as Sara. We'll show you Sara here in a minute, but there's a medium blending from the white lateral stripe to a darker green color. And that can be seen with the naked eye. And then if you look at Sara, it's really stark. That yellow you see above the white is not consistent in Sara, so I didn't use that yellow in addition to the white as something that was taxonomically relevant. But let me just show all this over again. This is Julia. I mean, this is thoosa. This is Sara. So that there's a picture of all three caterpillars. And as I went out of Utah to other states, I saw this consistency throughout the entire complex, except Northern California, Southwest Oregon, where the dark green Sara larvae started to look a little like thoosa, which concerned me. But not every character is airtight, and that was the exception. So when is using larval characters helpful? Well, when Anthocharis pseudothusa was named, I looked at the adult phenotypes and I thought, well, is that a thoosa? Is that a subspecies of the southwestern orange tip? This was named by George Austin. Or is this a subspecies of Sara? And looking at the adults, I thought, you know what? It could go either way. I just didn't know. So I went to the type locality of in Douglas County by Jackass Creek in Douglas County, Nevada, and the larvae were classic Sara, not thoosa. When Austin named the butterfly, he did name it Anthocharis pseudothusa. He was correct, but he also referenced thoosa as Anthocharis cerathusa. He wasn't splitting Sara from thoosa, and Oppler and I were splitting Sara from thoosa, but pseudothusa is a Sara. And here's a picture of Sara pseudothusa next to Sara and thoosa. Another question I had is I thought, well, Alaskansis has to be a Julia because directly south of it in the Pacific Northwest, both Flora and Sulphuris are a Julia, but I wasn't 100% certain. And so, again, thanks to Ken Phillip, who got me females. They laid eggs. I raised them. I took pictures of fifth instar larvae. Their larvae conformed to Julia as I expected. And here's some photos of comparing Julia alaskansis to Flora from Yakima, Washington. I have Flora sulphuris. I don't know if Yakima, Washington is actually Flora or sulphuris. You guys probably know better than I do. John does, where you have sulphuris, where you have Flora, and where they blend. And then off to the right, you have a caterpillar of Julia from Albany County, Wyoming. So when is using larval coloration of fifth instars not useful? for my research, I basically shot them 48 to 54 hours after they turned fifth instar. But a lot of marbles and orange tips, not just Sara complex, but Eucloini, they change color right before pupating. And so if you take a picture like 80 hours after they turn fifth instar, you'll notice Sara, thoosa, and Julia Browning, they're all dark green. I think in one of the books in the Pacific Northwest, I think David James wrote, I saw a picture of a Julia flora where it was very dark green. It was taken not within this window that I used for my research. So once fifth instars are 60 hours or older, it's difficult to tell them apart. Let's talk about pupal curvature and color. In layman's terms, the pupal cone more or less is erect, or does it bend back? In this picture here, I've got a Julia from Colorado where the cone bends back, and I've got a thoosa from Colorado or southwestern orange tip where it doesn't bend back nearly as much. And that turned out to be a really consistent discriminator, especially where these two species fly near each other or in sympatry. As a standalone character, pupal cone curvature is not consistent when you're talking about Sara orange tips. So here's some pupal cone curvatures of the different subspecies of Sara, and they're all over the place. So I kind of threw Sara out when talking about pupal cone curvature and color as having taxonomic implications. Sara gunderi, again, that's a subspecies from Catalina Island. Sara thoosa I've talked about from near Lake Tahoe, extreme western Nevada, getting into California. Sara's all over, with southern California to northern California, and basically in southwest Oregon. And Sara sempervirens has kind of a longer cone, but I just could not draw any taxonomic implications from using the pupal cones of Sara. For example, this is some pupal cones from Ash Creek in Siskiyou County. One is erect and the other is bent back. So, but in Utah, where browning eye flies near thoosa, you've got this pupal cone of the southwestern orange tip, which is more or less erect, and the Julia is bent back. If you compare the southwestern orange tip to sulphuris, the sulphuris pupa, also a Julia bends back, and so on and so forth. This is the Colorado. This is Anthocharis of Colorado compared to Julia prestanorum, and just lots of examples. Even within species, I noticed that, and this is helpful, and I'm really getting into the weeds here, but with southwestern orange tips, thoosa from Utah, the cone is erect or bent slightly forward, whereas the other three subspecies, it's erect. If you look at thoosa Colorado, it's bent back just a little bit. If you look at thoosa ingami, it's bent back just a little bit. So that's also helpful, and if you have more questions about this, I can answer that in the questions. So let's talk about number of overwintering cycles. What do I mean by an overwintering cycle? Basically, it means a year. Usually in nature, we're talking about orange tips that overwinter as pupa. They have one generation per year. So when a caterpillar feeds the pupa and the pupa enters hibernation, let's say in the first week of May when it's done feeding, it will be dormant until it'll stay as a pupa, stay in hibernation through the summer, through the fall, and through the winter, and emerge the next year. So usually an overwintering cycle is just 1 year. But for my research, I would actually fit 2 overwintering cycles into 1 year. And if this is a little confusing, ask me at the end and I'll try to explain it better, because I'm still trying to fight the clock and get this done in an hour. But you don't, when a swallowtail or a pyrrid forms a pupa, it tends to be a pupa at room temperature or outside May, June, July, August, September. They don't need 5 months of warmth before they overwinter as cold. So I know I'm not explaining this really well. I put it to you another way. If an orange tip pupates in May, I would leave it in room temperature for a month, and then I would give it another cold cycle if it didn't emerge. I would give it another cold cycle in June, July, August, September, and it would emerge in October. So I apologize if I'm not explaining that clearly, but I did notice, I noticed this first with anti-swallowtails. In Utah, anti-swallowtails only have one flight, and I was anxious to get some adults out because I had some of the black form. And so they pupated in May. I kept them in room temperature until June, and then I gave them a cold treatment, June, July, August, September, brought them out, and they emerged in October. They thought they didn't need a long summer before having 120 days of winter. And so to streamline my research for southwestern orange tips, which tend to overwinter for multiple years, I would fit in 2 overwintering cycles in 1 year. If you don't understand that, it's not critical. But that's basically what I did. So let me share with you a graph that I put together for my paper. So for the Sara orange tips, for the 4 subspecies, I ended up raising 123, and the average overwintering cycles was 1.41. So basically took between 1 year and 2 years to emerge. And the reason why I'm saying this is, in Washington, if you guys were to raise Julia orange tips and the larva forms a chrysalis, you overwinter it, it'll emerge in the spring. But in Utah and other xeric areas of Western North America, if the habitat, like the Great Basin, tends to be dry, and the Colorado Desert and the Mojave Deserts are even drier, if those regions don't have a wet winter, the pupae will not break diapause in the spring and will wait and will assess conditions another year. And if it's dry another year, they'll wait yet another year. So that was what I was trying to say before, is if you look at this graph, you see that total for Aethus, I raised 207, and the mean overwintering cycle is 2.83 overwintering cycles. Whereas if you look at all the subspecies of Julia, the average is 1.07 overwintering cycle. So what that means is Julia flies in a much more boreal, forested situation where it has wet winters, the pupae aren't as critical. Here comes spring and the butterfly emerges, but not so much with thoosa and Sara. So how is this relevant is when thoosa flies with Julia, and yet thoosa takes multiple years to emerge in nature, but Julia takes 1. So again, at the end, if you guys have any questions about this slide, if I'm not being clear, I'll be happy to answer those. Okay, so the interspecific contact zones is the last section of this presentation. And we'll go over this and talk about what happens when different members of Sara complex meet. And again, we're going to be focusing on that zone 2 I was talking before. Okay, the first one, the 6 regions that I mentioned in my paper, 1 through 6, 1 and 3, I studied at the McGuire Center. And basically, where Anthocharis Sara meets Anthocharis Julia Stella, you don't get a lot of intermediates between those 2 species, according to Davenport. And in California, Nevada, where Anthocharis Sara pseudothusa meets Julia Stella, it's the same thing. So I myself did not study zones 1 through 3. And in my paper, I'm simply referencing what other people are saying. So just to summarize this slide, there's been several reports, and Davenport noted that where Julia met Sara, he was not getting any intermediates. Either at the Bass Lake along Beasore Road, nor at Big Sandy Campground. He found Julia flying with Stella. Usually Stella would fly at higher elevations because it's more of a boreal insect, but he did find them flying together without intermediates, suggesting that they are different species. These are pictures taken from the Ken Davenport collection. When we talk about Southwest Oregon, that's where Anthocharis Sara enters into Southwest Oregon, but it also meets Anthocharis Julia sulphuris. And when I met with Andy, he talked about in his book about where Sara meets, he called it Sara Stella, meets Sara Stella at Klamath River Canyon. He says, Todd, you've got to go to Klamath River Canyon. And so in between 4 and 6 May of 2006, I met up with Andy and Gary Pearson, and I basically visited 4 places in Oregon. I want to focus on Klamath River Canyon because at the other Rogue River and mile marker 8, basically all I found there was Sara with no evidence of Julia. And at Waltz RV in Chiloquin, all I found was Julia with no evidence of Sara. But as you can see, circled Klamath River Canyon had evidence of both, as Andy told me. So for lack of time, I'm not going to talk a lot about Merlin Gillese Road in Josephine County, Oregon. Suffice it to say, we found caterpillars. I reared them through. They conformed to Sara, and only Sara was there. Was in the contact zone with Julia. Applegate Road in Jackson County, I also found caterpillars there. Those were also Sara. But Klamath River Canyon was interesting. When I went to Klamath River Canyon, I collected 4 females that laid eggs, and I also found eggs on the rock cresses. And all the eggs on the rock cresses conformed to Julia sulphuris, except one was Sara. The females I collected laid eggs, and all of those were Julia. So here's a picture of the variation. The middle column and the right column are the Julia sulphuris from Klamath River Canyon. And as you can see, the males vary a little bit. The discal cell bar gets thicker with some of them. And this is what Andy noted in his book, The Butterflies of Oregon. I kind of went over that kind of fast, but he said something is going on in Klamath River Canyon. He thought the hybridization of Sara with Julia sulphuris, which he called Sarastella, because he was using the older taxonomic interpretation, perplexed him. He didn't know why it was happening. And I didn't really solve the puzzle either, only I discovered that the Julia sulphuris was showing amazing echotypes. And the one Sara that you see on the left doesn't look a whole lot different than Julia. So I would consider them cryptic species, but the larvae from that location do suggest that Andy was right. Kudos to Andy. He said he thought 2 species were flying in Sympatry, and he was correct. Here's a picture of the most extreme example of Julia sulphuris from Klamath River Canyon on the left, which kind of looks like thoosa on the right. But if you look on the underside of the marbling, it's green versus more of an olive. And the larvae, again, conform to sulphuris. I'm not going to talk a lot about where pseudothusa and Julia stella meet, but I did see specimens at the McGuire Center, where Austin collected both taxa flying at Kingsbury Grade in Douglas County, Nevada, without seeing any intermediates. So we looked at a lot of specimens. We were either seeing Julia stella or Sara pseudothusa. And then Davenport also stated that in Mono County, California, he found both taxa flying together. Here's some pictures from the McGuire Center of some Alpine, California, Julia stella on the right, and Julia stella on the left, and then Julia stella on the right, sorry. So the last part of my presentation, I'll be talking about some of the work that I did in Utah, where thoosa met sulphuris or browning eye in Utah, and where thoosa Colorado met Julia neoprestanorum in that state of Colorado. So for lack of time, I found 4 locations where I found Sympatry. Well, I found 3. Raft River Range in extreme northwest Utah, the Book Cliffs and Huntington Canyon. And in the Stansbury Range, there was some photos of Julia, and they were sulphuris, not browning eye, that were taken years and years ago. But I will talk briefly about southwestern orange tips and Julia sulphuris flying in Sympatry in the Raft River Range. And this is just a close-up of the maps. These are the GPS. The red is the thoosa, green is the Julia sulphuris. This is what the habitat looks like in the Raft River Range. And I did rear some from this area, but these are wing-collected specimens, Julia sulphuris on the left and thoosa on the right. So the question I had is, are they interbreeding? Are there any intermediates? And I did collect this one specimen that I thought might be intermediate, but I couldn't prove it. So it's just a hypothetical. But I do believe them obviously to be different species because there were so many parental of both and they were flying together. Okay, I want to talk a little bit about number 5, which is where thoosa meets browning eye in Utah. And I talked about these separately, thoosa, southwestern orange tips, and Julia browning eye. I wrote a paper on that in 2010 where I shared my observations of how they behaved where they met. And basically, as I've said before, the larvae differ. I've already mentioned that. So I thought if the larvae differ, and if the pupae differ, because I've talked about the cone differences between Julia and thoosa in northern Utah, their habitat is different. I mean, Julia browning eye, this is Rock Canyon in Provo, and this is also in Utah County. This is the Ochre Mountains filled with juniper trees. And there's something special about southwestern orange tips and juniper trees in their habitat. The Julia browning eye, we're using this particular rock cress, Becerra perennens, and the thoosa also uses Becerra or rock crusts, but they also use pinnate tansy mustard. But one thing I noticed when I found them flying together in Juab County is that the males of Julia browningi would fly slowly in a ravine in search of females. But the thoosa males would patrol much faster. They would leave the ravine and find a juniper tree to look for a female that maybe recently emerged as it possibly pupated. So the male patrolling behavior was distinct when I saw them flying together. So with mutually exclusive distribution habitat preferences, how, where, and why do they fly together? And the answer is in the Wasatch, and I apologize if I'm probably getting too deep. I'm approaching the end of this presentation. In the Wasatch, you'll sometimes, this is Julia browningi distribution, you'll get these juniper trees that have invaded into the Wasatch Mountains, and then the southwestern orange tips will follow them because the mustards, the pinnate tansy mustards, takes refuge, the host plant of the southwestern orange tips. So that's why we find them together. It's the southwestern orange tips invading into the habitat of the Julia orange tips. This is a region that I studied them in 2009 and 2014. And these are the places where I studied Little Birch Creek. Here's some of the data that I have. I'm going over this quickly because I'm probably running out of time. There were, to summarize this slide, there were some possible intermediates where they're hybridizing. Here are some specimens that I actually pinned and shot in 2009. The ones in the middle are possible hybrids, but I wasn't satisfied that they were truly hybridizing. So in 2014, I sought out to collect gravid Julia browningi females flying in close proximity to southwestern orange tip males to see if possibly if I could rear them and see combined traits in the larvae or in the adults. So within the area in Juab County, these are the two canyons that I studied, and I ended up collecting 4 females of Julia orange tips flying in close proximity where the male thoosa and the male Julia were patrolling. And the bottom line is I collected 4 females. They laid eggs. I didn't get a lot of eggs per female because I didn't want to overextend myself. To make a long story short, 3 of the 4 Julia females, the caterpillars were Julia, the pupae were Julia, and the adults that he closed were Julia. But 1 female, that was a browningi, Julia browningi. The larvae were clearly Julia, the pupae were Julia, but when they emerged, they were hybrids. And so on the left here, you see my southwestern orange tips. On the right, you have Julia. And the middle ones are what 3 of the adults from the browningi female, the Julia female, look like. And those clearly, I mean, if you look on the middle row towards the bottom, you look at that female, is that southwestern? Looking to the left, is that Julia? No, I'm fairly convinced that even though they are different species, since I live close to this location, I was able to prove that they will occasionally hybridize. And so that's the story with where Julia and thoosa meet in Utah, which is pretty straightforward. I was able to prove hybridization. The next area, and I'm almost finished, is in Colorado. Well, if Utah is easy to separate Julia from southwestern orange tips from thoosa, it is really difficult in Colorado. On the left, we have Julia neoprestanorum. On the right, we have thoosa colorado. Look at those adults. Those are different species. And Jim Scott had reported where these species were flying in near sympatry. And I just wanted to see if I could find both species in one location. And to make a long story short, I failed. They came close to finding each other, but they didn't. These are the counties and the locations that I studied in extreme southwestern Colorado and northwestern New Mexico. And these are the locations. I know we're running out of time, so I'll just briefly talk about Summit Reservoir in Montezuma County. I collected some females, reared the larvae, but the larvae, the adults, I thought they were Julia. The habitat looked like Julia. I mean, this is a forested situation in Montezuma County. Elevation is 7,000 feet. I thought, oh, this is Julia country. And then I collected some females, and the larvae and the pupae were clearly thoosa. And that took me by surprise. And it took the pupae like 3 or 4 years to emerge, not one like Julia. 9 miles away in Target Tree Recreation Area, now I'm moving from west to east in La Plata County, Colorado. These are the adults that were reared where the larvae, the pupae, were clearly Julia. So within 9 miles of each other, in Montezuma County and La Plata County. So you can see, according to this graphic, I got Thoosa, Colorado, within 9 miles of Julia near Prestanorum. So I'm going to read this from my paper. The identification of both Summit Reservoir, A Thoosa, Colorado, and Target Tree Recreation Area, A Julia near Prestanorum, was further verified through examining fifth instars, pupal cone curvature, and the average number of overwintering cycles it took for both taxa to emerge under the same lab conditions. Summit Reservoir took 3.73 overwintering cycles, typical of Thoosa, for a sample size of 14, and Target Tree Recreation Area, Julia, took 1. I would overwinter the pupa, they would emerge. Here's the larvae, which is consistent with Julia. The pupae were consistent. I'm sorry. Here's the larvae, which were consistent for Julia, for Target Tree Recreation Area, contrasted to Summit Reservoir, where the larva was consistent. I mean, everything was consistent. It's just the adults. So I wanted to show these two areas what the adults look like. Going down into the weeds, and I'm about 5 minutes from being done here, so if you hold out with me, I know I've gone to a lot of detail. One way to differentiate the adults of Anthocharis thoosa Colorado from Anthocharis Julia Prestanorum is to focus on the orange tip on the ventral surface. If you look at the orange tip of this female on the left, of Thoosa Colorado, there are some black markings adjacent to the orange tip, right? Now look at Julia Prestanorum. There's hardly any black markings. Here's another set of females. Same thing. Thoosa has some black markings next to the orange tip. Julia doesn't. So even though these adults look alike, that was something that I noticed to separate Thoosa versus Julia adults, where I had more than just larvae, pupae, and overwintering cycles to separate these two species. But I just thought it was amazing how close they were. And I had similar experiences in Durango, where I found Julia and Thoosa flying within 3 miles of each other. And I won't go into all the details. I found Julia in Wildcat Canyon and Thoosa in Carbon. So 3 miles apart. But to make a long story short, I was not able to find both species flying in sympatry. Close, but no cigar. But why do Julia orange tips look so similar to southwestern orange tips in this region of Colorado, New Mexico? I have no idea, but I find it really intriguing. When I went to other areas, I won't spend a lot of time with this. I found some orange tips, and they were Julia at Lower Piedra Campground in Archuleta County, as well as near Dulce Lake in Rio Arriba County, New Mexico. So the conclusions are there's a combination of 3 species in the complex. And highlighted here says these taxonomic conclusions are further supported by unpublished mtDNA from Oppler. Nick Grishin did some DNA sequencing of a lot of Sara complex specimens that I sent him. And he said he acknowledges the arrangement of 3 distinct species taxa of Sara, Thoosa, and Julia. Portions of the barcodes from that paper show the Sara complex is presented here, figure 26, with permission from the authors. So in other words, when you look at the barcodes, it clearly separates Julia from Thoosa from Sara, which is, I wasn't able to use this 6th character in my paper. They says, no, you need to mention in the conclusion, oh, by the way, DNA supports this taxonomic arrangement. But I want to give special thanks to Nick Christian and Paul Oppler for letting me use their data before they published it. They finally did publish it last year for this paper. DNA barcodes reveal different speciation scenarios in 4 North American Anthocharis, of which I'm a junior author. And in that paper, they support my conclusions. And that basically is my presentation. I want to thank John for all of the help that he's given me over the years. Especially, I mean, I'm very fond of the Pacific Northwest. I spent my high school years in Everett. I didn't say that to lead off with. I want to give special thanks to John and all his help, Andy Warren, Caitlin Labar, Dave McCorkle, Robert Conley, Gary Pearson, Terry Stoddard, Abe Holman, Dave McNeese, Chris Guppy, and everybody else who contributed to this work, several from the Pacific Northwest. I'm grateful for the Prestons, and that concludes my presentation. I am open to questions.
That's absolutely wonderful, Todd. It's so interesting. It's not really surprising that it took a while to sort all this out, since the adult phenotypes are so similar, and it was only by looking at other characteristics that you were able to separate these 3 species. So yeah, kudos to you for such a wonderful piece of work.
Thank you. I probably put in too much information in all my slides, but I had a hard time shortening it out, so I'm sorry about that.
Okay, does anybody have comments or questions that they would like to make?
Toddler, Yakima is flora.
Okay, it looked like flora, but I thought since it's east of the Cascades, it's got to be sulfurous. But I mean, okay.
You know.
Any questions? I know I went over a lot of material.
Toodles, I just want to say how good it is to see you. Been missing
Good to see you.
And what a terrific talk. What a phenomenal amount of, don't apologize, a lot of detail, but we needed it. You put in an enormous amount of research, absolutely granular, as you say, which is what's necessary in order to have faith that this stuff really means something. And you had lots of duplication and reiteration, and you delivered us all from orange tip hell, let me tell you. So some changes were necessary, but we were waiting for it for years, and when it came, it was rich and wonderful. So thank you so much for your magnificent.
I was blown away by Colorado and New Mexico. It's like, why do these two taxa look so similar here? It's just, that still blows me away. I have no idea. Does anybody have any thoughts as to why? I mean, I thought Utah was intriguing, but they were so divergent. But going into Colorado, I was just going, what is on here? Why do they look alike? I still don't know.
Hey, Todd, we're Northwestern boys and girls.
Yeah.
Well, I've spent some time in Colorado. Anyway, it's all good.
That's true, but not down there. The only place I ever saw orange tips in Colorado when I was a kid was up around Mount Elbert, at the base of Mount Elbert, the highest mountain in the state. I remember going up there with John Justice and saying, wow, there's orange tips in Colorado.
Right. Yeah, when I was a kid in California, I saw one orange tip in 10 years. I thought it was because it was rare. I lived 3 miles from the Verdugo Mountains, and so sometimes you get these montane butterflies would be strays on my block. I didn't know that as a kid. So when I finally saw an orange tip, I thought they were super rare. I caught a male Sara, and I was just tickled pink. Wow, I caught an orange tip. These are so rare. Well, as an adult, I went back to Grand Park and found them flying everywhere. I had no idea. As a child, I had no idea of a concept of habitat.
Yeah.
I just thought they were super rare. So that's kind of what hooked me as a child on Sara orange tips in SoCal.
Hey, go on, Todd. You got to tell us your story about Lorquin's admirals in the Northwest.
Story about Lorquin with a fried brain like this? What do you want to know? I mean,
No, about your experience in Everett, right? The first one you ever saw, right? In your yard. Yeah.
I had some slides on, I spent 2 years in Everett at Cascade High School. And when we talk about the, man, my brain is fried. I saw Lorquin's admiral flying in my backyard in Everett, and I just went, because I was more familiar with Wiedemeyer's admirals than Lorquin's admirals, which is a paradox because born and raised in SoCal, but my grandparents introduced me to Wiedemeyer's admirals in southern Utah, and I never got to get Lorquin's admirals except once in Southern California. But I saw one in my backyard in Everett. I just went, I grabbed my net, which out of mothballs, and couldn't find it. And I walked all around the forest. I kept thinking, Washington State is forested. It's just like southern Utah. There's got to be all these butterflies. I'd walk around gorgeous forested habitat and wouldn't see any butterflies. As an adolescent, I kind of entered diapause at that point in my life but yeah, this Lorquin's admiral, subspecies Ilgi, showed up in my backyard. When I visited you, John, when BYU got lucky against UW, I went back to Silver Lake in Everett and found larvae of Lorquin's admirals, because even when I was a high schooler in Everett, I had no idea of habitat. I just thought these butterflies you see once in a lifetime were just that rare. So,
Todd, I expect when you saw that Lorquin's, and having been
what's that?
I expect that when you saw the Lorquin's, having been familiar with Wiedemeyer's in the past, and all of a sudden you saw this admiral with orange tips, you probably thought you found a hybrid between a Wiedemeyer and a Sara.
Yes. I remember when I was at the Pacific Northwest Lepidoptera workshop, I was talking to Andy Warren. I says, yeah, I love Sara orange tips. I love Lorquin's admirals. He says, all you like, Todd, is butterflies with orange on their tips.
Yeah.
And I said, that's nonsense. I think Phyllodes sonorensis is a trash bug. What's the common name for Phyllodes sonorensis? Sonora blue. Well, it's gorgeous, but it has orange on its tips. And I was trying to be funny with Andy. It's hard for me to have a sense of humor when my brain is fried.
So I see a couple-
John Richards and Carter have had their hands up for a while.
Yeah, I was just going there. So John, I see you're unmuted. Why don't you go ahead and it's open discussion, so just chime
Okay,
in.
good. I have a question on DNA barcoding because it fascinates me. So
Yes.
the DNA barcoding you showed separates the species, but has there been studies done to see if the barcodes are different in subspecies?
My understanding is no, and I'm not qualified to answer. JP, do you want to jump in or somebody else?
I
Total
can-
genome.
Go ahead.
Oh, go ahead. No, man, you go, James.
What I was going to say was it very much depends on which subspecies of which things you're looking at, because we all understand that subspecies, if they're truly allopatric and truly genetically, I mean, if you're calling them something different, then they're going to have some genetic differentiation of some kind. The issue is that the barcode is one tiny part of the mitochondrial DNA only. And so, whereas entire nuclear DNA may give you a much crisper picture of the difference, and there should be some difference even between subspecies, you may have no barcode difference where there are subspecific differences, and vice versa. You can sometimes have intergradation of barcode DNA, mitochondrial DNA, so that even within the same subspecies, you've got different barcodes. So I guess the take-home message is that barcodes are useful. They may tell you who's reasonably closely related to who, or if things are different, but they're not the end-all and say-all. That tools that we now have that allow you to analyze entire genomes if you can, or bigger pieces at least, parts of the nuclear DNA, give you a clearer picture. And of course, Nick Grishin is doing some of that, although some people, of course, have a lot to say about that as well. So anyway.
This would be a good time to jump in, Todd. You asked me what butterfly we named for June Preston.
Yes.
It was Todd, Paul Hammond and I published a review of the Mariposa copper butterfly a few years ago. And we looked at the entire species throughout its range. And we, in the end, named 9 new subspecies, which we did with great trepidation. But so far, they've all survived the Pelham test in the catalog. So we think we had some legs to stand on. We named most of those after women who have had a significant impact on Western butterfly studies, including June Preston. And one of them was also named for my late wife, Tia. But we named one Junia for June Preston in Western Washington. And it's one that we had found, but I went out with June and Floyd to show them that butterfly. And of course, they picked it out of the brush. So I thought that's a good one to name for June. Well, you mention that now because one of the other subspecies, we honored Felix Sperling with a patronym. We named it Lycaena or Tharsalia, Mariposa Sperlingi. Now, Felix is one of the great genomicists that we have working on the nuclear and full genome studies, as well as the mitochondrial. And Felix was, I think, pleased by that, but he was also intrigued by the entire Mariposa system. And we invited, Paul and I, at the end of our paper, said, we've done, like you, Todd, we've done this on traditional, we hope careful, phonetic grounds for the most part, though you use much more than just phonetics. And we did too. We used biogeography and a few other things. But we said we would invite anyone who is capable of testing these conclusions with the contemporary technology of genomics to do so. And Felix, bless his heart, took it up. And just this summer, Felix, in conjunction with staff from the Washington Department of Fish and Wildlife and the Xerces Society, including Candace Fallon, for whom another subspecies that she discovered was named, have been collecting the material for Felix's lab to look at exactly that, James, the nuclear DNA of subspecies in this case. So we might get a good answer for your
Nice.
question, at least for this system.
Well, Bob, can I share with you a story about June Preston? When she visited me in, it's right on my site, I think it was 2007. I showed her my collection. And back then I hadn't done a lot of work with Lycinity. And I showed her the ones that I had pinned in my collection. And I only had a drawer at the time. And she looked at me and she says, "Todd, that's all you got?" She got trashed me. It was so awesome. I said, "This is what a delightful lady." She says, "Yeah." And since then, I've done more work with Lycinity. But yeah, she called me out. It was funny as can be. I loved it. It's like, dude, you're a light.
That's classic. That's classic June. She did that sort of thing all the time. She was very blunt, straightforward. So.
Oh, but very sweet. I mean, I love June.
Yes,
so.
I'm not saying that she wasn't. Carter's been waiting to talk for a long time.
Yeah, but just before, in case people don't know who we're talking about, so it won't be an in thing, June and Floyd Preston collected all over the country for decades in order to provide a synoptic collection of North American butterflies that could then be studied forever. They mostly ended up at the McGuire Center, and they did more direct field work than maybe any other couple in North American butterfly history. Excuse me for butting in with that. I want people to know who the Prestons were.
Okay. I want to butt in too. I want to butt in right now about Floyd and June. They were mentors to everybody. You talk to people from coast to coast, Ken Phillip in Alaska, anywhere you go, that longtime people, Floyd and June, been there with the arms around them. I'm telling you, young people, old people, it doesn't matter. Floyd and June were everybody's mentor. And you always learn something, or you got stimulated. Is that all you got?
Okay, thank you for those lovely tributes to the Prestons. Carter, you have been waiting for a long time. Would you like to pipe up?
This is all fascinating and exactly what I was going to ask about. And I would invite James and anybody else to chip in on this. But Todd, this was such an exhaustive dive into the life history of these species, and I was also going to ask about, maybe to put you more on the spot, but today, if you were going to start this over again, would you be looking like Felix, maybe more at straight up, more sequencing more of the genome in order to answer these questions? Or do you think that there's still a place for these life history characteristics, even if in 5 years it's cheap and easy to sequence the entire genome, as it seems like it's getting cheaper and easier every single year?
I'm going to leave the sequencing of genomes to other specialists because I don't have that expertise. For example, right now I'm really into Euphilodes blues, trying to separate Euphilodes Bowers blues from Euphilodes stanfordorum, but there's no larval differences, and so I'm kind of frustrated because I don't know how to do genomic sequencing or to counter what Nick Rishin is doing, say he's right here, he's wrong there. I can only study these where I have competencies. And for a lot of, I have noticed with Euphydrus anesia complex, the anesia checker spots, some interesting larval characters, but where there's no larval characters, the life history studies aren't as helpful. And as one person, I can only do what I can do.
Carter, as a partial answer to your question, I think what Todd has done is show that careful field work is absolutely essential, because sure, you might be able to characterize something looking at the DNA, but you don't know anything about its actual biology, how that DNA translates into what's actually expressed in nature. And so I think when Todd does something like he's done, and then afterwards the barcodes bear him out, that's just reconfirming that doing the biology is critical.
Right on, James.
That's what it says to me.
Right on.
I would also add that being an old geezer, not as old as you, Bob, being an old geezer has allowed me to see the evolution of various, techniques and approaches to butterfly systematics. And I don't know if anybody remembers back when barcodes first came into the community of scientists and systematics. They was the thing. They was the bomb. Everybody barcode this, barcode that, barcode. Well, they're still useful, but they kind of been passed. And now it's a total genome. But like there's proteomics, and there's transcriptomics. Like there's a whole bunch of things that's waiting for us. I don't know if anybody ever looked at thumb drives. You got a thumb drive, 15 years ago, and you had what, maybe half a megabyte on it. You get a thumb drive now, it's got a terabyte on it. That's how much technology is advanced, and genomic technology is keeping a pace. So I would say that genomics is going to get to the point where Todd will have his what's that Star Trek thing that you got when you can tell anything by pointing at it?
Tricorder.
Tricorder. Yeah, we're going to have tricorders, and it might not be in my lifetime, but it's around the corner.
Yeah,
we never thought we'd have Dick Tracy watches, huh?
We're still going to need natural history even then.
Yeah,
yes,
no matter what.
Absolutely, and
It's a.
I wouldn't want anybody to think that I was
right.
going again. I mean, I've been a field biologist my whole career. I wonder about. It sounds like, and I completely agree that the barcoding and the sequencing just falls in with all that other life history information, because you have to know how the organism actually lives its life.
I look at it as an extra set of data that you could tap into, not a replacement set of data you tap into.
Right.
And that's what Andy told me. He says use as many character sets as you can.
Yeah, exactly. Because that's what we're talking about here. We're talking about different character sets that are hopefully tightly related to one another. So.
Right.
And it helps when you have.
Sorry?
It helps when you have people who have the extraordinary discriminatory powers to see the subtleties of those character sets. And we do have a number of them among us. James Scott was one. Andy certainly is. Jonathan Pelham is. And people who have a good eye. And Paul Hammond is another. Without Paul, I certainly never could have completed that copper study, because I'm moderately observant as a naturalist. Maybe I aspire toward that. But Paul has one of those remarkable, almost preternatural eyes to see those distinctions. And Todd, I think you've got that, and you need it for those euphalotes you're working on. So good luck with that.
Mm-hmm.
Yeah.
Well, the other thing. Oh, sorry. Shut up, Todd. I'm going to talk good about you. So the whole thing about us in the Northwest. I keep telling everybody about James and Nunley, their life history book. That dedication to life history is so important, and it's abundantly obvious in that book. It's abundantly obvious in this presentation, Todd. How much caterpillar poop did you have to clean? How many pieces of litter did you have to empty? All of that work. That's dedication. That's love, brother.
Thank you. I appreciate that.
Mm-hmm.
So there's a question, a very specific question, Todd. Patricia, do you want to unmute and ask your question about the overwintering cycles?
Yeah, please do, because I was not very clear about that.
Patricia?
Well,
there you go.
Yeah, I forgot my question already.
So
the Sara orange tips are in the desert. I just wondered how many overwintering cycles or whatever they go through. I live in the Pacific Northwest, right? So I'm used to the 1 year deal. Right. And then you were talking about the dry location.
I don't know if I raised a lot of Sara. Sara flies, excuse me, in Alonzo Borrego, barely getting into Imperial County. I didn't rear those. They were clearly Sara. I do know that thoosa ingami, I'm remembering from 3 or 4 years tops. The desert orange tips, however, would go between 3 and 11 years. And that wasn't that was an actual year. I wasn't cheating with overwintering cycles, trying to fit 2 into 1 year. And so it's a desert orange tip that shows the most best adaptability to the desert, where they went between 3 to 11 years before they would emerge.
Yeah. 11 years, a decade?
Uh-huh.
Yeah. I remember Jack Wolf said I'm not raising Anthocharis, thoosa, puma anymore because if I get pupa, they're gonna outlive me.
I've heard
I
of-
was gonna ask Todd if you had a maximum length of time that you had noticed overwintering, and I take it 11 is your maximum. that you've
11.
had.
Yeah, I had two left, two thoosa pupae left. One died and one emerged a healthy adult.
Mhm.
So 11 seems to be. But these things that fly in the desert are so equipped for the desert. It's just
Yep.
amazing.
Well, it surprises me no more. It used to surprise me a lot when things that are adapted to habitats like that can last, variable numbers of years and often into long periods of time. And even those things that we have in the East, like Lunas, it was funny. I have this story about opening a drawer at the University of Kansas when I was a graduate student there, and because I heard this rustling in there, and I opened a drawer and there's this live Luna moth in a cocoon in the drawer. And I went around asking people, how long has this been there? And it had to have been at least 5 or 6 years is what I got. And it could have been longer. And so you probably have heard stories about what are called pupal banks. The idea being that the way things make it through extended long droughts like they are in the Southwest is the fact that not everything dies off. You've just got things sitting there in pupa waiting, and they can wait for quite some period of time. So.
Yeah. The butterfly's ability to survive is just blows me away.
Amazing.
Nature never stops teaching is something I try to live life by. I mean, nature just, that's why you do field work. Nature always teaches you something new. When you think you've got things figured out, you don't.
Yeah.
I think John Emmel wrote about Indra swallowtails going 10 and 12 years and more.
Mm-hmm.
And the ones that I've raised, because Indra, I was almost into Indra more than orange tips, but Indra, the most I've had is 5 years. But the thing about Indra is their host plants are perennials. And so they don't have to struggle in the desert as much as desert orange tips because their host plants are annuals. And if the Mojave Desert doesn't get the right amount of moisture between December 15th and February 1st or whatever it is, their mustards won't germinate. And if their host plants aren't up at all, then the pupae just waits a year, waits another year. Whereas Indra swallowtails, even during dry, it's a perennial. It'll have some host plant growth where maybe a few Indra might fly, but it's the orange, desert orange tips are even worse shaped than Indra swallowtails.
Yeah.
Is there any information about what the cues are, whether it's chemical cues or temperature, humidity, or what causes?
I only have field observations for desert orange tips. It's the right amount of moisture during a time window, but I haven't solidified it down. I haven't specified it, but it has to be increasing day length, and there had to have been a certain amount of precipitation during a certain time window. And they are aligned with, there's 3 mustards that desert orange tips use in the Mojave Desert of near Mesquite, Nevada. I won't go into their names, but and I've looked for them in certain years where the host plant just doesn't break ground, doesn't come up. And the pupae know that. The butterfly is in sync with its host plants.
Senior Stout. I think I told you this. I got a pupa one time that Dan Carney gave me because he thought it was dead, right? It was Indra.
Yeah.
And it seemed heavy to me. So, I kept it. I actually, it had a girdle on it. So I stuck a pin through the girdle and, kind of artificially placed it in a cup. And I poured water on it. 2 weeks later, it emerged. That's after 3 years.
Yeah. I think the actual cues they need to break hibernation is far more sophisticated than what I'm saying. It seems like I keep saying, oh, we've got nature figured out. We don't. But the more we spend time in nature, the more we learn, but we'll never figure it all out. It's really complicated and amazing.
Well, I'm really disappointed. I can't imagine that we're not going to figure it out.
So many species, so little time.
Any other questions? This has been fun. This is funner than the actual presentation because I can interact. I hate lecturing. I'd rather interact with people.
You're so good at it, Todd. You're so good with kids.
Thank you.
Even old kids like us.
Yeah,
Yeah,
right.
I was going to say, us kids too.
We're all kids 96 and under. So.
Okay, any other questions, comments? There are a couple of comments in the chat, just commending your incredible hard work, your great photography, and especially your detective work, Todd.
Thank you. I hope it wasn't too much information. I worry about that, but
That was
Gotta
marvelous.
have the complete story.
Yeah, we got the story, exactly.
yeah.
Okay, well, thank you so much, Todd, for joining us.
Okay.
And, next month's meeting, we will have Bob Pyle. And he will be, are you going to be playing your recording, Bob, or?
Yes, yes, I was asked to give a lecture to the Royal Entomological Society last spring, I think it was, and it was recorded by them. And it's a lot more work to give it again. So I will introduce it live, but from then I will share my screen for the recorded presentation. It's called 50 Years of, it's called From Monkswood to Hollywood, 50 Years of Butterfly Conservation.
Nice.
Please join us.
Yeah,
Okay.
we'll look forward to that, Bob. Okay, well, thank you so much, everybody. Thank you, Todd, for joining us. We
Okay.
really
Thank you. It's good to talk to you guys.
Todd, so good to
Good
see
seeing
you.
everybody.
thank you.
Good night.
Hey,
Thank
Good
you,
night.
Todd.
See y'all next month.
Bye-bye. Thank you, Regina. I mean, Paulette.
Another
Regina. Thank you, everybody.
Okay, good night, everybody.